Is there a way to turn it on and off? I don't think I did the right thing.
It should just, it's not,
Not turning off.
I can turn just the lamp off, and then we can just set everything to,
Okay.
like turn laptop, and then, when you're ready to use it, I can turn the lamp back on and, reroute it.
Okay, thank you. Good morning.
I might have to leave early because of work. Is that okay?
Yeah, sure.
Okay.
No problem.
And I might come back, but it's,
Yeah, don't worry about
yeah,
it.
thank you.
So before we start the lecture, I think I left something out during the last lecture when we talk about the one sample hypothesis testing for the proportions. We have a z-test, and we also have an exact test from R. For the two sample hypothesis testing, instead of one sample, I said that aside from the normal approximation, there is also an exact test. So that exact test is actually the Fisher's exact test that we'll talk about today. So if you want to take a note on that, these two lectures are related in the test for the proportion. I think it was the test function. So for the this test function in the last lecture, this is also using the normal approximation. If you want to have a exact test, you will use the Fisher's exact test, and the input will be a little different. I guess I will just start. Today we'll talk about paired testing and some tests for categorical data, as well as non-parametric tests. So we'll talk about two sample paired tests, when to use it, when not to use it, what is the benefit and disadvantage. And we'll talk about distribution-free tests. By distribution-free, we mean that we do not assume the data to follow a certain specific distribution, but there is still requirement on the distribution for the test that we're going to introduce. And then we'll talk about the inference for contingency tables, including the chi-square test and the Fisher's exact test, and also odds ratio, risk ratio. So let's first go to the pair data. In our last lecture, when we were talking about two-sample tests, I briefly mentioned that if you are able to sample two groups that are paired with each other, you are able to eliminate some of the variance in the data, so you will have more power in the testing. So today we're going to formally introduce the paired two-sample t-test. Suppose that you want to compare two sunscreens, and you want to know which one works better. You can do two experiments. The first one, you can sample two people, and then randomly assign them into two groups, one receiving sunscreen A, one receiving sunscreen B. And in another design, you only have to sample just half of that sample size, and you put one sunscreen on one arm, the other sunscreen on the other arm. And you can immediately see the advantage of this, because you eliminate the possibility that when you compare the left arm with the right arm, you eliminate the possibility, the variance that come from comparing different people. So you would be able to directly see the difference in one people, and that improve the power of you detecting that difference. And there are many other settings. For example, we have some examples down here. For example, the same person before and after treatment. You can also do 2 times the sample size and look at their before and after treatment. These two scenarios, they sometimes one of them is not possible depending on your study design, the nature of the study. For example, some people, you are probably only able to do a before treatment sampling, and after treatment, you are not able to do that, and vice versa. So in those situations, you probably can only do the unpaired testing. But as long as you are able to do the before and after, then those will improve your power of detecting that difference. And when you have measurement of the same sample twice, 3 times, that is also those two samples will be correlated with each other. And if you want to compare the first measurement with the second measurement, for example, trying to decide how stable your sequencing process is by comparing the sequencing result of the first measurement and the second measurement, then that will also be a paired testing. Same measurements on twins, same measurements from 2 body parts, for example, taking the nasal sample and the oral samples, those will also be paired. And matched case control studies at the subject level, this is also sometimes considered as paired. So a matched case control study is a study where it's a retrospective study where you look at a set of samples and you try to understand whether a risk factor is related to an outcome. So what you will do is you separate the subjects based on their outcome disease, outcome that is healthy, and then you would choose one patient from each group and try to match them based on the other covariates. And then after you match those people into pairs, you can compare their risk factor. So the reason why you do this is because when you match those people by their covariates, you sort of have twins, and that improve your power of detecting the influence of the risk factors. So that is the case control studies where we can sometimes use pair testing. So you can see it's actually pretty common. There are some benefits of doing this. So if you have 2 samples, if you are a pair of samples, are very correlated with each other. One implicates a lot of information about another, then using paired testing would give you a lot of advantage, a bigger power. But if they're actually not very related, if you do a super bad job matching pairs in your case control studies, then doing paired testing may not help you. Because later on, I will show you the mathematics formula, the comparison of the unpaired test and the paired test, and you will actually see that when there's no correlation between the pairs, using the paired testing will actually make it a little harder to reject because you look at the larger critical value to compare the t-test statistics with. So that's one benefit and drawback depending on whether your samples in the pair are correlated or not. If they do correlate, then you have the advantage of saving the sample size. Instead of doing double the sample size, you cut it by half. But another drawback of pair testing is that if you have one person that drop out, you immediately lose two samples. But with the unpaired test, you only lose one sample. And another thing that you need to make sure of is when you take the paired measurements, they need to be measuring the same variable. You cannot be measuring the, for example, the LDL before treatment and HDL after treatment. You have to measure two of the same variables. If you measure different variables, your paired testing will have an entirely different meaning. So be careful what you put into the hypothesis. What are the interpretations of your hypothesis? Sorry. So now let's go to the definition. So I'm going to use the one something to denote the first group and the two something to denote the second group. Those observations will each follow a different normal distribution with different mean and different variance. You don't have to require them to have the same variance at all. And the way to do it is actually really intuitive. You take the difference between the pair, left arm minus the right arm, and then you just test if this difference, you test this difference against zero. It doesn't matter whether their variance is different or not because you are always able to calculate the variance term of this difference. And that is contributed by the variance of each group, and also you can see this row here is the correlation between the pairs. And since after taking the difference, we only have one set of values, we can just use the one sample t-test and compare this set of values, the mean of this distribution with zero. Null hypothesis would be the difference being zero, which is the same as the two means being the same with each other. So using the knowledge we learned from the one sample test, you calculate the mean of the difference, the standard, the variance of the difference, and you construct a z-test based on that, or a t-test, depending on your sample size. And here, the sample size is not the sample size of the two groups. It's the number of pairs. In a paired test, your sample size is the number of pairs. So here, I want to emphasize that the philosophy of this pair test is you compute the difference between the pairs and then treat them as single samples, and then you do it using the one sample t-test. There's no need to estimate each one of those components, the sigma 1 squared, the variance of the two groups, the correlation, those do not need to be estimated for this test because all of them are built into this sample variance. You can see that we're assuming the difference to be a normal distribution or approximately normal. That doesn't mean that your original y, they are also normally distributed. As long as their difference is normal, you can use this test. And if you want to check for normality, you check the normality of this difference, not the original one. So here we have an example. Still, we're using the LDL from beginning of study and end of study. We want to see if that change is different from 0. We actually did the same test in the last lecture, where we took the difference as the measurement and use one sample test comparing that with 0. That is exactly the same as treating the beginning time point as one set, treating the end time as another set, and doing 2 sample pair test. So you will see that the result is exactly the same. And you can use t-test with paired, with x as the beginning, y as the, do I have 2? So here, this is the way that we did in the last lecture. We take the difference and we use unpaired test, one sample t-test, because there's only difference here. There's nothing for the other group. But if you want to use paired test, you would put beginning and end at each one of them. It doesn't matter the order, and you would use paired equal to true for the paired test, and they will have the same result. Any questions? And you can also use formula. It's really versatile. So when you have a data frame, you don't have to subset one column by their treatment group. You can just use a regression type of syntax where you put the measurement, the values on the left and your treatment groups on the right, and it will give you the same result. So that's the paired test. When we do power calculations for the pair test, like I said, our sample size is actually the number of pairs. And another factor that comes into play is the covariance. So in the power calculation formula, you will need to plug in this sigma d. This is the variance of the standard error of the difference. And in the standard error of the difference in that formula, up here, this will be sigma d squared. This sigma d squared has the variance of each group and also the rho, the correlation between the pairs. So when you plug in the sigma d, you need to have a sense of what is the correlation between your pairs, or if you have previous knowledge about just the variance of the difference. In reality, we don't really know. When we don't really know what to expect in the value of this correlation, we would calculate the sample size for a range of rho. So you can calculate the sample size by plugging in different values for the rho and different values of this sigma d to give you a range of sample size. Another thing that I want to mention is that when you expect no correlation between the pairs, it's actually better if you do it unpaired, because when you look at the test mathematically, so here, this is the test that we have for the paired test. And for the 2 sample test, sample t-test, this is the formula for the equal variance 2 sample test for simplicity. Sample testing. The t-test here. So if you compare the two test statistics, they both have the difference in the, where is the test statistics? Yeah. So if you look at the two test statistics, why does it have to zoom out? So here, in the numerator, in the paired test, we have the difference. This is actually the same as the unpaired test. In the denominator, you have a variance estimate of the difference. That's also similar. They're not the same, but they're similar. They're estimating the same thing. But if you look at the degree of freedom for the test, for the unpaired test, your degree of freedom is the sample size of the two groups. So that's counting the number of subjects instead of number of pairs. But if you look at the paired test, the degree of freedom is actually the number of pairs. So an argument is that if you have no correlation between the pair and you use paired test, it's essentially the same as doing an unpaired test with half of the sample size. So you would want to use the paired test only when you are sure the correlation is going to help you. So that's all about the paired test. Any questions? And the paired test is still a t-test. It has distribution assumption on the observed data. And when you have a large sample size, it helps you to relax the distribution assumption. But when you have a smaller sample size, you would want to rely a little less on the large sample assumption. And that's when the distribution test, the non-parametric test, come into place. Non-parametric means that you don't need to have a distribution that assume parameters on the mean, on the variance, on the shape. But it's not assumption free. So later on, we'll actually describe two tests that assume your data to be symmetric at least. And the non-parametric methods can be applied when you only have ranking instead of continuous measurement. So if you have, for example, in questionnaires, you have 1, 2, 3, 4, 5 categories, and you cannot really say if the difference between 1 and 2 is the same difference as the difference between 2 and 3. Those situations, it's easier to use. It's actually more valid to use non-parametric testing instead of treating the 1 to 5 as continuous measurement. However, when you do have a continuous value and you do have a normally distributed measurement, using the non-parametric test will lose some power because you are wasting information on the distribution of the data. If you know the distribution, use it. If you don't, then use a non-parametric test. They are also very good for small sample size. When you have a small sample size, it's hard to see the distribution shape. The non-parametric tests are safer, although you also lose some power, but it's easier. It's actually safer doing it in the non-parametric way instead of drawing a wrong conclusion based on a distribution-based test. And sometimes when your data distribution is not normal, it doesn't mean that non-normal immediately takes you to the non-parametric test. You can first try to do some transformation of the data and see if you can make it normal. And if you cannot, then come to the non-parametric test. So knowing your data, knowing whether it's ordinal data, categorical data, continuous data, looking at the distribution of the data, and see if you can transform it. Those are the things that you need to do before you choose the test. We'll introduce two non-parametric tests. One is for one sample testing, and the other one is for two sample testing. And just like we did for the pair testing for the pair t-test, the one sample test or the pair test, two sample tests, the pair test can be transformed into a one sample test. So we'll just talk about one. The assumption is that suppose it assumes that the observations come from the same continuous distribution. We do not generally use the non-parametric test on count data because the assumption here is the distribution is relatively symmetric. If you have count data, most of the times you will have a large tail on the right. And that means the symmetrical assumption here is validated, so normally we don't really apply this directly to the count data. So the assumption is that under the null hypothesis, this will be the mean, the center, the median, the center of the distribution is around a certain value. So here, suppose the null hypothesis is M0, then suppose the null hypothesis is M0, it means that under the null, the distribution center, the median, is at M0, and the alternative is whether the distribution is different, the center is different from M0. And regardless of this, the value of the M0, you can subtract that from your data, so your data will be centered, and then you can test your center data against 0. And that's why we simplify this test into just looking at the median equal to 0. The whole procedure of the test is related to the order, the rank of the data. Suppose after centering, you have all these values, centered values, and you would rank them from the smallest to the largest. The test itself discards all of the 0 values in the later calculation, and then you rank all of the non-0 values by their absolute value. You take the absolute value of those measurements, rank them from the smallest to the largest, and then you also keep another vector that save the signs of those values. So you would rank them based on the absolute value and keep record of the signs of the values. And intuitively, if the distribution is symmetric, so here is one example. The measurement is the change, and we order them by absolute values. What is the smallest absolute value is this one. And then it's the next one is here, and this one, regardless of the sign, you rank it in that way. The largest absolute value is here. And you keep another column for the signs. Intuitively, if your distribution is symmetric around 0, then you would expect the signs to be evenly distributed across the ranks. Because if you have more values that are positive, you would have higher ranks that are more positive signed. If your distribution is more to the negative side, you will have more of the lower ranks that have the negative signs. So you would want the signs to be roughly equally distributed, and that is the philosophy of this test. So you take the test statistic is actually take the number of positive values and the rank of so you take the sum of all of the ranks for sign equal to positive. You sum up the 7 plus 3 plus 10 plus 6 plus 13 and 14 and 15. That will be your test statistic. And another test statistic is to sum up all of the negative ranks, but they are actually just two sides of the same thing. You use either one, you will have the same test, same result, because the summation of all of the ranks, this is going to be a known number. So the summation of the positive ranks or the negative ranks can either one of them can be used as the test statistic. And the distribution of this is more complicated. So if you do it by hand in the old days, you will be provided with a table of critical values to compare with. And now we have the computer, so we can use the Wilcoxon test with one of the input as the difference or the one sample that you collected. And you can also choose to test the median against 0 or some other values. You can choose the confidence level and whether to plot the confidence interval or not, output the confidence interval or not. So for this data set, if you sum up the positive ranks like I did, it will be this one. If you sum up the negative ranks, it will be this one. And regardless of what you use, you will have the same p-value. Any questions? So you can see that the whole philosophy of this test is to see if more values appears on the right or on the left. And that really requires your distribution to be symmetric. If you have unsymmetric distribution, then by ranking them and comparing them to the 0, you actually would not, even if your median is at 0, you will not have the correct p-value. So the Wilcoxon rank sum test, this is a parallel of the t-test in the non-parametric world. Like I said, if you know the distribution is normal, using t-test will be more powerful. If you use the non-parametric test, then you lose about 5% of the power to detect the difference. This is actually not a lot if you're really worried about the non-normality. So non-parametric test is actually very commonly used, especially for small sample size. And it is also robust to outliers because remember, we only assume it to be a symmetric shape, but it doesn't assume the tail to shrink like the normal distribution. So if you have super large extreme values on each side, it doesn't really affect how you rank the data. So that's why it is robust to the outliers. But for t-test, if you have fatter tails, then your mean is fine, but your variance is not fine. So t-test will be very sensitive to the outliers, especially when you are in small sample size. In large sample size, the outliers effect kind of get washed out by the sample size. So one thing you want to keep in mind is that you're assuming the distribution to be symmetric. That's the one sample test. And we also have two sample tests that's called the rank sum test. The other one is signed rank test. I often misspeak the two names, Wilcoxon, because they're both named after Wilcoxon. This one has another name, Mann-Whitney U test. They are exactly the same test, just named after two different statisticians. And so this one is a two sample test. And the assumptions are, first, you have independent samples. And the second one is that the variable of interest is also continuous because you will actually be comparing the overlap of the two distributions. For count data, that is not very meaningful. So the model assumption is that you have two distributions, each set of samples, and you want to know if the probability, if the two distributions they overlap, if you randomly draw two samples from one sample from each group, if they are the same distribution, then the chance of one sample being larger than the other one, that should be just a flip of coin, right? When you randomly choose two samples from the same distribution, the probability of one larger than the other one, that should be just a flip of coin. So under the null hypothesis, if you take one sample from treatment A, one sample from treatment B, then the probability of each one of them being larger, that should be half. That's the indication of this test. If you have two distributions that are slightly different, and you draw randomly draw one sample from one of them, then the larger distribution will have a larger possibility of giving you the larger value, right? So can you guys see this? So when you randomly draw two samples, one from this shape, one from this shape, then the probability of the value from this shape larger than the value drawn from here, that probability is not a flip of coin anymore. It's larger than 0.5. And the test itself is essentially trying to see whether the values drawn from this distribution ranks higher in general than the values drawn from this distribution. So the way we are going to rank it is you would mix up the two set of values and then rank all of them from the smallest to the largest. And if you have more group A on the top of the rank, then that means your group A has smaller values. If you have more group B on the bottom of the list, then that means your group A has larger values. But how many is many? Because when you randomly draw the samples, there's some chance that your group A will, on average, rank a little higher than your group B. So after you rank the data and calculating the rank for group A, you would compare it with the distribution and see if that is actually a significantly higher rank or lower rank. So the test statistic is after you mix up the two samples, rank them from the smallest to the largest, you calculate the sum of ranks for one sample. And you calculate the sum of ranks for the other sample. And you can use either one of them to construct your test statistic, because those two sum up, they will be a constant, known constant. So this one is your test statistic, and you would compare it with a table, or you use R to calculate the significance of the test. It will be easier if you look at the data in context. So here we have this data set again, but in the first one sample test, we ignore the arm. We're just trying to see if this difference is from 0 or not, right? We didn't really look at the arm column. For the second test, for the two sample test, we're going to separate these changes into two arms and compare arm B with arm C. In that situation, you would rank them based on the find value instead of the absolute value. You rank them. So unfortunately, I should have put the data on the slide. So let's do some hand calculations here. What is going to be the rank for them if they are ranked by the signed value instead of absolute value. So the first sample, maybe this can be a little exercise for you for this part of the lecture. So let's rank them. What is going to be the largest one? That's still the bottom, 15. And then that's the 14. We have 15 samples. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. This is the observation number. And we're going to, I guess here, and we'll rank them. This is 15, 14, and this is 13. What is the next one? After 42, it's going to be, the next positive value is this one, 6. And 11. And then 10. What is the next positive? This one? Right? Is that right? And what is the next one? After 2, it's going to be negative 2. And
So this is like the smallest negative number next to the order.
oh, right. So it should be here, right? This is 8 and this is 7. And then the next negative number is this one. And this one. So I'm just ranking all these numbers. I'm ranking them from the smallest to the largest. The rank on the table here, on this table, this is the rank based on the absolute value of the change. And now I'm doing a rank of the signed value without taking the absolute value. Because early on when we try to rank them by the absolute value, we're trying to see if this vector is centered around 0 or not. Now we're trying to see if the C group is going to have larger value than the B group. So let me try to finish rank those 4, 5, 4, 5, 12. This is 7. Sorry.
Oh, yeah.
And then 6.
If 5 is negative 1.2.
Oh, this one is between. So this is 5. This is 4, 5, 6.
I treated and they did on Excel.
Right. It's just hard to copy the data here.
Right, I had.
Let me see if I have the data in R. Okay, it's here. Oh, so it saved us all the effort. So now I'm going to rank this data frame by the order of the change. This is so this is after the ranking. I ranked it by the change value, and I want to see if one of the arm ranks higher than the other arm. If you have one arm, for example, the B arm, having much smaller value than the C arm, then you would expect the B's to appear more here and the C appear more here. So if you sum up the rank for the C group, if you sum up the rank here, these values for the C rows, you would expect that sum to be much larger than the for the B group. So that's the philosophy of this test. Well, thankfully R saved my day. But you can imagine in the old days or in the undergraduate test, you will actually be ranking numbers by hand. I will probably fail my own undergraduate test. Do you guys have understand what I'm talking about for this test now?
So like when you say that the rank for C would be higher than B, like how would you
Quantify that?
like so you were saying if like B and C are different, then the rank for B would be higher than C.
Or the other way.
They'd be really the difference
Yeah.
would be large.
Yeah.
How do you determine that difference is large enough to like call it?
Right. So that's the table that the table of critical values that you look up. Yeah. But that's a really good question. When you calculate the ranks, for example, when you have 4 data points and you see B, B, and C, C, is that a significant test? Or when you see 10 values and you see all the top 5 in B and all bottom 5 in C, those two will have different significance levels. And this is just for 2 groups, and you can imagine this being done for multiple groups. For example, when you have ABC arms, you can calculate the sum of rank for each one of them and decide whether that rank is too high or it's still within the reasonable range based on randomness. It's actually related to the enrichment analysis for gene expression. I'm not sure whether any of you heard about the GSEA enrichment analysis. So in those situations, you have the gene expression fold change for all of the genes. Suppose you have 1,000 genes, you have the fold change for all of them based on your data. And you want to know if a certain pathway, a certain subset of the genes are more highly, have more fold change than the background. So this will be your enrichment set, the enrichment pathways. And the way to do it is actually the same. You rank all of the fold change and you see if this subset of gene ranks a lot higher than the background gene. Any questions? And I imagine you can do this for microbiome too. When you have functions of the genes, you can rank the effect size. It doesn't have to be fold change. It can be anything that indicates the significance of the effect. So shall we take a brief break after talking about this test? So here we have the Mann-Whitney U-test. We have the function to do it in R. It's actually the same function as the one sample test, but you have two inputs. You can also use the regression format, which is the way I'm doing it here. So the W test statistic here, I think it's not the sum of rank for one group. It's actually the test statistic that's calculated this way. So it has the mean subtracted. This is actually the rank of the expected rank of group one if group one and group two are randomly mixed up. So when you calculate the rank of one group, you subtract the expected value of that if your data is randomly mixed up. Your sample size for each group will actually also decide which one of the group has larger ranks. For example, here in our example, we have roughly the same number of C and Bs, but suppose you have fewer Bs and more Cs, this test is still valid. The test is going to account for the number of the imbalanced number sample size between the two groups and compare the sum of rank with the fair value. So that's all about this test. We'll take a break and come back at 10, and I will stay for questions.
Yeah, we should quick question.
Yeah.
So are you still assuming symmetry for the two distributions?
For this case, it's not symmetric, but it needs to be continuous.
Okay.
Yeah.
But yeah, by the way, I'm the PA for the next 3 weeks.
I barely recognize you.
Yeah.
question.
Mhm.
So in the RNA-seq data, when you're calculating of all the change, are you calculating it based on the relative abundance of
Yeah,
gene expression?
yeah. So that's why sometimes we use the test statistic as the input for enrichment analysis because
Okay.
that accounts for the variation as well.
So for those used to use the raw values, I don't understand what's happening in RNA-seq data. Like when you're calculating fold change,
Mhm.
what do you input to calculate the fold change? Is it like the
Um,
absolute, like gene expression values, or is it the like number of reads, or is it a proportional number of reads?
For the RNAs, it depends on the software.
Okay.
For DEC, the input is the raw count.
Okay.
It actually try to estimate the sequencing depth.
Okay, and then it'll just
Yeah,
store it
yeah.
magically.
It's actually so
Interesting.
the distribution assumption is a negative binomial distribution, and that has the
That
total
makes sense.
count in there.
Yeah.
For some other method, I think for Lima, I'm not super familiar, but I think the model assumption is more a mixed effect model. So for that input, you can actually use continuous values as well.
Okay, so I should be careful about whether I'm putting in raw or proportional
Right.
data depending on the test I use.
Yeah.
Okay,
Mhm.
thank you. But if we have to like compare the microbiome data, what do we do? Like if I have lung microbiome and gut microbiome from the same mouse, what do I do?
Well, do you want to compare gut with lung because they can be really different?
I mean, I think that would be a good study to check because I think in the lung microbiome field, in mice, people still ask like, is it different from fecal microbiome when they're breathing in and eating their poop? So I feel like that would be a good study to check. But how would I go about doing that?
I actually have a paper on that.
Okay.
So my test is also pair test. It's based on dirichlet multinomial distribution. So it also counts for the count and over dispersion.
Okay.
And I actually gave some freedom to the distribution, so it's not strictly dirichlet multinomial. So for dirichlet multinomial, the outer layer is a multinomial, and on top of that, inside that you have a hierarchical. You
have lost me.
Okay. So I will stop there. But for my test, we test it at each taxonomic level of the tree.
Okay.
And in the end, you will have a global p-value telling you whether those two groups are different or not. And you will also be able to tell which part of the tree gives you that significance.
I've noticed that some of your older papers you use MATLAB. Is that?
No, that's one.
That one in R or MATLAB?
That one is in R.
Okay, good.
Yeah.
Thank you.
The one that I use MATLAB for is because MATLAB has a very good optimization package back then. I think right now R probably has has improved.
Yeah.
But I just didn't have the chance to
Yeah, I think it was your longitudinal one, and then I read something and I was like, oh, MATLAB. I don't remember how to use it.
Longitudinal?
Something along those lines. Oh no, it was the
regression.
Multiple linear regression.
Yeah, that one. That one should really be written in Python or R. But back then I didn't know better.
No.
I was just trying to push out the paper.
Great, thank you.
Yeah, no problem. So now we're going to talk about contingency tables. This is a situation when you have two variables each are categorical, and you are trying to assess if two categorical variables are associated with each other. For example, if your exposure to a certain risk factor is associated with your outcome, and your outcome is disease or non-disease, responding to treatment or not responding to treatment, it has to be categorical in both variables. You can do it for ordinal variables as well. The category 1, 2, 3, 4, 5, rating 1, 2, 3, 4, 5, those can also be put into contingency tables. But when you use contingency tables, you assume those 1, 2, 3, 4, 5, their order doesn't matter anymore. Does that make sense? So you're in your test, if you switch the label for 2 and 3, it will be the same. Mm-hmm. Right. There is something called the ordinal logistic regression. So logistic regression is just like a more flexible method for categorical data. And what you actually do is actually choose one of the categories as, for example, the baseline, the level 1, rating 1 as the baseline, and you will calculate the effect size of each of the categories with respect to the first one. And then you also have some assumptions about how those other categories, relative to the first level, those effects are also interconnected with some parameters. But the philosophy is that you choose one group as the reference and compare the other ones with this group. And you assume that those effect is also ordinal. When you increase from 2 to 1 to 3 to 1 to 4 to 1, there's some assumption in the parameter. But if you do it with the contingency table, it doesn't matter. Your 1 and 2 is reversed. So you have categorical data, and you will tabulate the samples into a table. This table, the row size, the number of rows and number of columns, those are not your sample size. The sample size is the summation of count in this table. So for example, if you have 100 samples, and half of them are treated, half of them are control, and then you observe some of them responding, some of them not responding, you will tabulate it into a 2x2 table, each cell categorizing whether it's responding under the treatment, non-responding under the treatment, responding under control, non-responding under control. Those 4 categories will be the 4 cells of your data table. Your sample size is not 4. Your sample size is 100. And for contingency table, we'll talk about 2 tests. One is the chi-square test, which requires the sample size to be relatively large. And we'll also introduce the Fisher's exact test, which doesn't assume, it can deal with a table where you have small counts in the cells. But when you have a large table, when you have a large count, the Fisher's exact test can crash in R because it's going to do a lot of calculations in the backstage. And when you have a large count, the chi-square test is actually not that bad. So you can choose between those 2 tests depending on your sample size, the table size, and the number of counts in each cell. So here I have an example for a larger contingency table. Your treatment doesn't have to be 2 levels. You can have more than 2 treatment groups and more than 2 outcomes. And you just tabulate all of them into this table. The total of the counts is going to be your sample size. And after you tabulate, I just have to. So see here, you can assign a variable to the rows and columns arbitrarily. So switching the order of the row and column, you will have the same table. It doesn't matter. So depending on what your research question is, you can do a chi-square test of homogeneity or independence or goodness of fit based on this contingency table. The first two are equivalent. The third one is also equivalent in the sense that you're comparing your count with a known distribution, but it is actually very magical that the first two are actually the same, exactly the same in terms of the test, in terms of the model assumptions and the under the null. So the test of homogeneity, this is a test where you want to see if this column has the same distribution as this column and this column. So if you observed pure level 1, you would also want to observe pure level 1 here and here. The homogeneity test is trying to see if the distribution of the variable 1 is going to be the same for all the values of variable 2. If you're, let's put it into context. It's trying to test if responding to a treatment is the same for treatment A and B. So that's homogeneity, right? Your distribution, your outcome, distribution of the outcome is the same in the treatment A and treatment B. That's the test for homogeneity. And that's actually just another way of saying independence. Your outcome is independent of the treatment because your distribution of outcome is the same for each treatment group. So the homogeneity and independence, they're talking about the same thing. But when you interpret the test based on your data, one of them may click better with the general audience. So when you have this contingency table and you get the chi-square test, you get a significant p-value. You can either interpret it in terms of homogeneity or independence, depending on which one is more meaningful in your, is more easier to understand in your context. Does that make sense? So when you have two variables that are, when you have a 2x2 table, exposure, non-exposure, outcome, dead or not dead, it's probably easier to interpret it in terms of the independence. Being dead or not is irrelevant, is independent of your treatment. But when you have, for example, treatment, non-treatment, and your response is, I'm satisfied, slightly satisfied, not satisfied, very dissatisfied. In that situation, it is probably easier to interpret it as a distribution, like a homogeneity, whether your the distribution of your response is the same between the two groups. Does that make sense? Yeah. So it really depends on which one is easier to explain. So the third one is slightly different from the first two. When you only have one sample and you want to see if when you only have one group of samples, for example, suppose you only have one row of them, and you want to see if this distribution is different from, for example, all of the p's equal to the same value. So suppose you have just one treatment and you have 1, 2, A, B, C, D, E, F, 6 values for the response. And you would have all these counts here. And you want to see if all those are even. Your null hypothesis would be PA equal to PB equal to all the way to PX. Or your null hypothesis can be PA is actually half of P. Those type of hypotheses can also be tested using a chi-square test. But the derivation and the test using R will be slightly more complicated. I'm just trying to let you know that there are situations where you use the chi-square test to test the distribution of a categorical variable against a null hypothesis, and the null hypothesis is the cell probability, the probability of falling into each of the cells, if that is following certain pattern. Any questions? So if you have categorical variable, one group, and you want to see if one of the categories have larger probability, or if the probability of getting those categories are the same, then you can also use a chi-square test. The philosophy of all of these tests is actually the same. So it's actually going to look at the count of these tables and compare to what you would expect to observe in the contingency table under the null hypothesis. If your null hypothesis is that all of the columns are have the same distribution, you would expect the count of those columns to be similar, and that will be you will actually compare your observed value with the expected value. If those two values are very different, you reject the null. So let's try to. So I think I just talked about the homogeneity, the independence, and the goodness of fit. You can look at the formula here, but I think I'd rather not repeat them by reading the slides again. When you have a 2x2 table, this is the most common situation. This is the one that I wrote before the lecture. I don't know why sometimes it's appearing from the center or on the side. So when you have a 2x2 table, the test of independence is actually when you calculate the probability of exposure, which is those two sum up, and the probability of case, which is those two sum up. The probability of case multiplied by probability of exposure, those two should be the same as this cell. So that's the independence assumption. When it's independent, you multiply the probability of one event by the other event, it's going to be the probability of those events joined together. That's the independence assumption. And under the independence assumption, you have this one, those two multiplied to this, and those two multiplied to this, those equal to this, and those multiplied to this. And we're also going to assume all of the four sum up to one. And if you solve this second equation, you are actually going to get the same result as this. The ratio of those two is going to be the same. So the distribution of case is going to be the same as the distribution of control. So it's easier to see independence and homogeneity are equivalent if you look at the 2x2. That's basically what I try to, what the slides try to say here. The testing procedure is easier. Why? Oh. I think I'm done with this whole thing. So this is a 2x2 table. We'll use it as an example. And suppose this is the observed value of the 2x2 table. The 0 and 1s, they indicate the two levels for the exposure and the outcome. And we'll use the O to denote them because those are the observed values. We'll also calculate what are expected under the null hypothesis. So what are expected? Under the null hypothesis, you would expect the two rows to have the same probability, homogeneity, or independence. So you would expect the total count to be distributed into those categories based on those probabilities. Suppose that for variable 1, this is for you. Suppose that you have N1 under the treatment and N2 under the control, and those subjects should be distributed into responding or not responding based on the same probability. Does that make sense? This is your treatment group. You would expect them to be distributed between responding and non-responding based on this pi probability, and this pi probability will be the same for treatment and control. This is what you expect under the null hypothesis, and you would calculate the count you would expect under this null. So you just plug in the pi as the overall proportion of non-responding. This pi will just be the value here. The pi will just be the value here over the total value, because that's a proportion falling into this non-responding category. You plug in pi equal to this over this, and you will have a table of expected values under the null. This is the expected number of patients falling into each cell under the null hypothesis. It may not be an integer, but this theoretical value should be close to your observed value under the null hypothesis. So the test statistic would be the difference of the observed values and this value, and you will sum them up. The test is going to be, so this is the expected count. It doesn't have to be integer, and you try to assess if this is different from the observed table here. That's why we calculate the test statistic by taking the difference and sum of squares. And you also want to balance the weight you put in each cell by divided by the expected count. You don't want because when you have more count in one cell, the difference is also going to be more in that cell. You want to balance that out. That is the test statistic, and it's going to have a chi-square distribution. The degree of freedom depends on the number of categories. So if you have a study where you can categorize the outcome into 3 categories instead of 5 categories, your degree of freedom for the test for the three categories, that will actually be smaller. And if you look at the distribution of the chi-square test, the larger degree of freedom is, the more larger values you will have in the distribution, and that means it's harder to reject if you compare the same test statistic with the chi-square distribution of different degree of freedom. It will be harder to reject when you have a larger degree of freedom. So in some situations, you may not want to have too many categories. Grouping them into more meaningful, smaller number of categories will probably give you more power to detect that association. So you would want to reject when this difference, the chi-square statistic is larger. You compare it with the chi-square distribution of the corresponding degree of freedom, and then you will be able to get the p-value based on all these chi-square distribution related distribution, the functions in R, but the easier way is just to use the chi-square test. And this X input, it should be data table, that's 2 by 2, the 2 counts, or you can put in the 2 columns of your data frame, where one column is for exposure, one column is for the outcome. So it's still very versatile. You can use the regression format where you use the outcome, wiggle, and exposure, or you can tabulate your variable into a 2 by 2 table and put it in there. In some situations, you may not have the 2 columns. Some collaborator just give you a 2 by 2 table that's already tabulated. That is still, you can still handle that using the same function. So here we're doing it in 2 ways. One is the 2 by 2 table. One is the data frame, 2 columns. Questions? So this whole test is under the assumption that the test statistic is going to be approximately chi-square distribution. The chi-square distribution, the F distribution, the T distribution, those are all closely related to normal distribution. So if you in your research, you see an F test, T test, chi-square test, those have normal assumption behind it. You either have to have a large sample size or you need to have a normal distribution somewhere. But when you have a very small count in cells, this approximation by chi-square distribution is no longer very accurate. So that's when you would want to test using the Fisher's exact test. This test can be used when your sample size is small, when certain cell when your event probability is rare. So you may have 1,000 samples, but if you have a very low probability of observing a certain outcome, of observing death, for example, then you may want to use the Fisher's exact test because the cell of one of the cell in your 2 by 2 table may be too small. The rule of thumb is that if your sample size is between 20 and 40, then you should have all of the cell count to be larger than 5. If one of them is smaller than 5, you should use the Fisher's exact test. If they're larger than 5, then you can use the chi-square test. If your sample size, the total count is larger than 40, then it's tolerable. It's okay if you have one of the cells having a very low count. But just like I mentioned, it's tempting to use Fisher's exact test all the time because you think that it's more accurate. But when you have a large contingency table, it may crash or throw in an error because it's going to do a lot of calculation to get you that p-value. Our software needs to go over all of those possibilities to calculate p-value for your test statistic. So the Fisher test can also be done using the same syntax, either 2 by 2 table or the 2 columns of the data frame. But the Fisher's exact test and the chi-square test, they are just 2 different tests that test the same hypothesis. There's no difference between the hypothesis that they do. It's just, it depends on the sample size that you have, whether you have small count in the cell of the table. Any questions? So the contingency table can be multiple rows by multiple columns, but the 2 by 2 table is the most commonly seen contingency table. And for 2 by 2 tables, we also have some other measures to assess the association. We have the risk ratio and odds ratio. So we'll also talk about that for 2x2 tables. Here, I would like to introduce a few concepts for study designs. We already talked about the risk factors, for example, exposure to certain treatment. We also have a difference between cohort study and case control study. So the difference between those two is that for cohort studies, they're mostly prospective. And you are able to have samples with and without the risk factor identified, and you want to see if the outcome developed for each of the exposed groups. For case control studies, it's mostly retrospective. You already have a set of samples, and you want to see if the distribution of the risk factor is different or not between certain outcomes. Those two are not the same, mainly because one is prospective, you have the treatment and you observe the outcome. One is retrospective, when you already have the data and you try to see what are the distributions of the risk factor and the outcome. We also have a difference between cross-sectional study and longitudinal study. Cross-sectional studies when you have measurements from one time point. Longitudinal study is when you measure the same variable over and over again for the same individual. And normally we would call a longitudinal study when you have more than 2 time points. For 2 time points, it's a little hard to use the existing longitudinal method, but cross-sectional study only has 1 time point. And you also have there are also experimental control studies where you are able to manipulate the treatment. This is more common in mice study, where you can control all of the risk factors of the animals and give certain animals treatment and observe the outcome. But for human studies, they are mostly case control and cohort studies. The reason why we talk about this is because risk ratio and odds ratio, they have different applications for these studies. So let's go to the definition of the risk ratio and odds ratio. When you have a 2x2 table, you have outcome, you have exposure, non-exposure. The 2x2 table will let you calculate the probability of outcome given you're exposed and the probability of outcome given you're not exposed. It's basically dividing the estimation for these probabilities will just be dividing the number of outcome and exposed over the number of exposed. So your null hypothesis, your point of interest is to compare whether the probability of an outcome is different between the 2 groups of exposure. And you can compare those 2 probabilities in many ways. You can take their difference, you can take their ratio, and you can also take the ratio of their odds. So what is an odd? Odds is the is the ratio of outcome for the exposed, given they're exposed, divided by the probability of not the other outcome given they're exposed. It's more commonly it arises from the gambling world when you try to if you gamble, if you play bet on horses or games, they are mostly in terms of the odds. You have a probability of winning, you have a probability of losing, and you divide the probability of winning by the losing. You don't just look at the probability of winning. It's more intuitive in that situation. It also has a better mathematical property compared to the risk ratio. Or actually, so when I say the probability of winning and losing, that's for one outcome, and you can calculate that odds for another outcome. For example, winning and losing, if it's a rainy day, and you will have an odd. And the winning versus losing on a sunny day, that's another odds, and you will compare those odds by taking their ratio. That will be the odds ratio. You should be careful about interpreting odds ratio because it's tempting to say, for example, when the odds ratio is larger than 1, that means the probability of happening, the first event happening is this pi 1 is going to be larger. Well, so the odds of the event 1 is going to be larger than odds of the event 2, but that is not equivalent to the probability of the event 1 is the same as the probability of the event 2. So this ratio equal to larger than 1 or smaller than 1 is different from this ratio is larger than 1 or equal to 1. So it's you would want to avoid saying one event is more likely when you have an odds ratio that's, well, so this ratio is different from this ratio. When you have an odds ratio that's 2, you shouldn't say that the first event is twice likely than the second event, because that's actually interpreting this ratio. Does that make sense? So try not to interpret the odds as likely, because likely means probability in statistics. So the risk ratio, the risk difference odds ratio, those are alternative ways to compare two distributions, two binary distributions, probability having 1 or 0. If you use the risk difference, this is actually the same as what we did in the two-sample testing with proportion. The risk ratio and the odds ratio are new concepts, so we'll develop different tests for each one of them. So if you try to decide between risk ratio and the odds ratio, here are some considerations. So the odds ratio is a little hard to conceptualize because you're taking the ratio of odds, and odds is already a ratio. So it's a ratio of ratio, and it's a little hard to interpret. The risk ratio is easier to interpret, but on the other hand, the odds ratio is actually more mathematically robust, and it will also be applicable to both retrospective and prospective studies. But risk ratio is only applicable when you have prospective studies. So here, when we try to interpret those things under the null hypothesis, if your risk factor exposure is independent of outcome, then you would expect the risk difference to be 0, the risk ratio to be 1, and the odds ratio to be 1. Those three are equivalent because they all mean that pi 1 is equal to pi 2. But the interpretation of the values will be different for the risk ratio and the odds ratio. If you have a risk ratio of 2, it means that event 1 is twice likely than event 2. And if your risk ratio is half, it means event 2 is twice likely than event 1. But if you have odds ratio that are 2 or half, you cannot say that one event is twice likely than the other one. You need to say that the odds of one event is doubled. So it's not more likely, it's the odds that's doubled. Here I highlighted on these. Don't say likely when you use odds ratios. And why is risk ratio not applicable to case control studies? Why is it only used in prospective ones? Because in case control studies, we sampled based on the outcome. We sampled based on whether they are dead or not, and then we see how many of them are exposed in each group. In a prospective study, you sample based on exposure. You have one exposed group, one non-exposed group, and you look at the distribution of the outcome. Does that make sense? In case control studies, you first choose your responders and non-responders. You choose the two groups based on the outcome, and you check whether those two groups have the same distribution exposure, the same distribution of risk factor. In prospective studies, you separate your two groups based on the exposure, and you check if the outcome are the same or not. And it's actually the latter that's more meaningful, whether your outcome is the same for the two different exposures. So if you calculate the probability of outcome based on exposure, so the risk ratio, this reversed. So what we want to when you have a risk ratio, you when you have a case control study, this is the risk ratio that you calculate: the probability of exposed in the outcome and the probability of exposed in the no outcome. This is the risk ratio that you calculate. But what you're actually interested in is the probability of outcome based on exposure. And those two quantities, they are not the same in case control studies. So that's why in case control studies, we would want to use odds ratio, because you can actually write out the odds ratios. So well, I will just save you from the mathematical calculation, but the algebra. But if you calculate if you write out the odds ratio in terms of these probabilities, you will find out that it doesn't matter whether you have exposure and outcome behind this bar or before this bar. It will actually be equivalent. So odds ratio can be used for both case control studies and prospective cohort studies. This is one thing that you would want to be very careful about, and that's why odds ratio is actually more commonly seen in the literature. Any questions that you would want to ask? So the risk, the risk-related estimators, they are mostly cohort cross-sectional studies. The odds ratios, they can be both cohort and case control. Another benefit of the odds ratio is that it's actually also related to the logistic regression that we'll talk about next time, or maybe in the next lecture. Because in your logistic regression model setting, the effect size is actually trying to estimate the odds ratio instead of the risk ratio. So you would see only the odds ratio in the interpretation of logistic regression. And here, let's try to calculate the risk difference for the 2x2 tables. For this table here, the risk ratio, the risk difference is the probability. So this is N11 over N1 plus. That's the probability of having the outcome in group 1. And this is the estimated probability of having the outcome in group 2, exposure group 2. And you take the difference. And then the confidence interval can also be constructed using their corresponding standard errors. And this is the same as what we did for the two sample tests for proportion. There's one drawback about risk difference is that when you take the difference of these two, for example, you have a very low probability in both groups, then that difference is going to be treated in the same way as the difference when you have a high probability in both groups. And in this example, the two probabilities in the two groups, they are very close with each other. Even if you take the ratio, they are still relatively the same. In this situation, the probability of the two groups, the difference is very small, but the ratio is high. The probability of your event happening is actually 10 times in one group than the other one. So when you take the risk difference, you ignore the baseline effect size. It can be misleading if you use the risk difference because in reality you may have a 10 times risk, but your difference is very small. So this is not a good statistic if you have very rare event. And for the risk ratio, you just take the estimations ratios and the confidence interval. We also have a walled type of, so when you have plus minus the normal quantile 1.96, this is also called a walled type of statistic. Walled is the WALD is a name for a statistician. When you see the plus minus Z, plus minus T, those are walled type of statistic and confidence intervals. The walled statistic assume the distribution of the test statistic to be normally distributed. So that's why you have a Z here, and it has the underlying assumption that you have a large sample size. So the risk ratio will take into account of the baseline. When you have very rare event, it's still able to give you a fair assessment. The odds ratio and the odds. So the odds ratio is going to be actually the diagonal element multiplied together divided by the off-diagonal elements multiplied together. And it doesn't matter how you arrange the rows and columns. If you have if you shuffle the rows and columns, you will have an odds ratio that's inversed. So having an odds ratio of 2, or 1/2 is going to you need to be careful about which is in the denominator and which is in the numerator. Which one is in the first column and second column? Because the calculation here, the numerator and the denominator, those will be reversed if you switch row 1 and row 2 or column 1 and column 2. So you may have an odds ratio of exposure over non-exposure, that's 2. And the odds ratio of non-exposure over exposure, that's 1/2. Those are the same thing. And your confidence interval reflects that because you have all of the cells in the denominator here. So that is actually regardless of where your rows and columns are positioned. So the odds ratio is can be approximated by the risk ratio when you have a very, very rare event. Because if you look at the odds, if your pi 1 is very, very low, then 1 minus pi 1 is going to be very close to 1, right? If both pi 1 and pi 2 are very rare events, then those two values will basically cancel out, right? If you have 0.001, 0.002 here, those ratio, the pi 1 over pi 2 will be very different, but the 1 minus pi 1, 1 minus pi 2 will be quite similar. So the odds ratio is going to be very close to the risk ratio if you have rare events in both sides. So here in this calculation, we're showing that no matter whether you have a prospective study or the retrospective study, odds ratio is safe because if you calculate the exposure before the bar, outcome after the bar, you will have the same value. It will be N11, N12, the diagonal elements in the numerator, the off-diagonal elements in the denominator. But if you look at the risk ratio, it's not, your rows and the columns are treated differently because you are taking the row sum, the column sum in the denominator here, and the column sum in the denominator here. If you switch the row and columns like the case control study instead of the cohort study, then you will have a different risk ratio. Does that make sense? So the risk ratio calculation, in the calculation of risk ratio, the exposure and the outcome, those two cannot be switched. In the calculation of odds ratio, the role of the two variables do not matter. You are just calculating the association. It doesn't matter if it's conditional on exposure, the probability of outcome, or the conditional on outcome, the probability of exposure. The risk ratio, the two variables are treated differently. The odds ratio, the row variable and the column variables, they're equivalent. And if you want to do it using R, you can use a library called epitools, where two functions, the risk ratio WALT and the odds ratio WALT, will give you the estimation and confidence intervals of the ratios. And the interpretation of the output is a little tricky. So for the risk ratio WALT, it will take the first row and the first column in the denominators. So here in your denominator, this will be the test will put the first row in the denominator, and or the first column. It's actually just the first row, not first column. It's just the first row in the denominator. So you can see in the predictor row here, in this output, there's nothing for the first row. It will be the risk ratio of the exposed 2 over 1. So if you calculate the risk ratio here, it will be 4 over 115. 4 over 119, and divided by 32 over 199. In the mathematical formula I gave you guys, you have the second row in the denominator, but unfortunately for this software, it's the first row that's in the denominator. And for the odds ratio, it also have the first row and column in the denominator. So when you interpret it, you also need to be careful about which one of them is in the first row and column if you use this software to get the odds ratio estimate. So what are we going to say about this specific this specific data set. So let's first look at the risk ratio. The risk ratio is the risk of disease 2 in the exposed 2 over exposed 1. That means that under exposure 2, the risk of getting disease 2 is going to be 1/5 of the same risk in exposure 1. That's how we interpret it. The risk of having disease 2 is 1/5 compared to the exposure 1. For the odds ratio, the odds of having disease 2 in exposure 2 is also roughly 1/5 compared to the exposure 1 group. You can see how close the odds ratio is with the risk ratio because the disease 2 is a relatively rare event here. And of course, you can do it by hand. But I'm glad that the computer is taking over all of our lives. And when I don't remember some of these concepts, I can even ask ChatGPT for the answer. So if you don't remember which one is good for which, ask ChatGPT. Can risk ratio be used for case control studies? It gives you the correct answer. I tried. And that's all I want to cover for today's lecture. Any questions? I think it's pretty amazing that ChatGPT is taking over a lot of the roles of instructors. So education is no longer about delivering knowledge anymore because you can get all those knowledge by asking the robot and searching online. It's actually, education is more meaningful in the sense that it inspires your creative thinking, critical thinking, and improves your logic. Right now, fortunately, ChatGPT is not able to do creative or logical thinking. So I still have a job. Well, I will stay for some more time. You're free to stay for more questions.
That's a good question.
I'm learning Categorical microbiome data. So this data and metadata and I did some analysis like this. I would like to analyze metabolic data. So I see the PLC and PM.
Metabolomic data?
Yeah. I have no idea. idea how to integrate the metabolomic data to the,
Hello-seq?
yeah, because,
I don't think, oh, well, you can use it as a meta table.
so I read the exercise and the main frame and
So you're reading in the sheet in one table, natural data frame. You can put it into the metadata of the phenotype object. So here, yeah, in sample data, you have metadata here, right? In this